Well, welcome to Lecture 4 in Social Neuroscience. And what we're going to talk about today is more about the nervous system. The last lecture covered the neurons, neurotransmitters, how neurons communicate with one another. But today what we'll do is focus really just entirely on the brain. And in order to do that, I want to keep in mind that I'm going to be making references to Sapolsky, and Sapolsky has this great book that sort of accompanies these lectures called Behave. And so you'll see that much of what I'm going to talk about here is in the second chapter of Sapolsky's book. So exactly what are we going to cover in this lecture? Well, first of all, we'll have an introduction to MacLean's Triune brain. It's been around for 50, 60 years. Then we're going to go ahead and break it down and look at the brain in terms of these three layers that are talked about in the triune brain: the reptilian or brainstem areas, the limbic system, and the neocortex. And finally, we'll end the lecture by looking at the specific example of how these layers communicate with one another by looking at how emotions and social behaviors go together. All right, so before I go on and talk about the triune brain, I do want to make a disclaimer here, and Sapolsky talks about this a little bit in his book, but I just want you to know that the whole idea of the triune brain is really considered passé. It's not anything that's really adopted these days. There's a lot of problems with it. You can see here's a 2020 article by Cesario and his colleagues in which he says, your brain is not an onion with a tiny reptile inside. And you can see further, there's a quote down there, this belief, although widely shared and stated as fact in psychology textbooks, lacks any foundation in evolutionary biology. So I'm about to tell you about something that's usually considered discredited, doesn't really follow what science says these days, but in a moment I'll show you why we're going to talk about it. But anyway, what did MacLean actually say? So Paul MacLean's triune brain model, which is discussed in chapter 2, posits that we have three layers of our brain, and one layer is known as the reptilian brain. In MacLean's way of thinking about it, he thought of this as the oldest part of the brain in terms of evolution, that this would be shared with almost any other animal on the planet. They all have something like a reptilian brain, and it would remediate the autonomic or automatic regulatory functions of the body, and it's found in the brainstem and the basal ganglia. Layer 2 would be known as the limbic system, and it's something that's maybe known more with mammals, according to MacLean, and this would largely mediate emotion. And then layer 3 would be the neocortex, this big part of the brain in humans and other primates, but also, of course, found in other mammals, that mediates higher cognition. And so this is, like I said, found in the cortical regions of the brain. Now, I said to you that there is some criticism of this, and Sapolsky does mention what some of the criticisms of this triune brain model is. First of all, it does seem to make it sound like we have these three very distinct layers, but there is actually a lot of anatomical overlap between what these three layers are doing. In reality, the flow of the information can go up or down. Originally, it looked like maybe the neocortex would feed information to the limbic system would feed information into the reptilian brain, but in reality, it really goes back and forth across the brain, up or down. In the third way of thinking about this is just, again, with the example of emotion. Automatic aspects of behavior, emotion, and thought are not really separable. You're going to see that we can't really just say, for instance, emotions only in the limbic system, that's where it's all done. These things about behavior, emotion, and thought are all very interrelated, so they're not quite separable the way the triune brain talked about it. The fourth criticism would be that the model mistakenly implies that each layer was added through evolution without any changes happening in the others. Like your reptilian brain is the same as any other reptile out there who has the reptilian brain. But in fact, as evolution has progressed with neuroanatomy, the three different layers actually are quite different and have changed over time, so that you don't see just like we all have a lizard brain inside of us. It's identical to what you would see in a reptile. And 5, the model mistakenly implies that evolution happens in some sort of linear progression, when in fact we have this sort of common ancestor model. And the idea would be here that it's not just like, well, you know, we were all once dolphins or sharks, and then we moved on to having mammal brains, and then mammal brains went on to human brains. This sort of linear progression just didn't really happen that way. It's much more of a family tree that you might want to think about, where there's a lot of different breaks and so on. So we don't really want to think or imply that somehow the reptilian brain came first, then we have this limbic system, and then finally the neocortex, that it happened in some sort of linear fashion like this. So despite all of these criticisms of the triune brain, you might think, well, why are we even talking about it here? And why does Sapolsky talk about it? Well, the reason why we're going to bring it up here is it's kind of a nice model of or a metaphor of neuronal organization. So if you really want to understand how the brain is sort of organized, it's sort of nice to think about it as like a metaphor about these three different layers. So keep in mind these caveats about the triune model, and that we're just going to use the triune model as just a way of sort of organizing our information about the brain. So let's briefly talk about layer one. This would be, in the nomenclature of the triune model, the reptilian brain or the brainstem. And I don't have a whole lot to say here because the brainstem would involve things like the medulla, the pons, the midbrain. These are like where low-level vegetative behaviors and reflexes are mediated. And so all that autonomic stuff that just kind of keeps us alive at all time in the background is largely done here in level one. And people can live in comas with just this activity going on. So you don't really need to have levels 2 and 3 in order to stay alive in the sense of, if you were in a coma or something. So level 1, it's important, of course, it happens to affect a lot of the things that we do with our bodies, but for the purposes of social neuroscience, we can kind of skip ahead now to level 2 or to layer 2. So layer 2 is the limbic system. And some of the main parts of the limbic system would include the amygdala, the hippocampus, the septum, the hypothalamus, the ventral tegmentum area, and the nucleus accumbens. And I don't have it on here, but parts of the prefrontal cortex, which we'll talk about a little bit later in this lecture. So let's go through these first 5 parts of the limbic system in a little bit more detail. So let's take the amygdala first. So the amygdala is important in fear and other emotions. It's likely to be involved in complex emotions such as trustworthiness, guilt, and embarrassment. And it has a central role in aggression and male sexual motivation. So amygdala, as being part of the limbic system, is quite important, as you can imagine, for lots of different kinds of emotional behaviors. It's very important and has been brought up a lot in the last 25 years in social neuroscience for understanding our human social behaviors. And so I'll just give you a little bit of an example of this. For instance, if we just take the amygdala, look at that and say, what if we find a patient who has a lesion to their amygdala? So there's been some sort of damage just to their amygdala, but nothing else. It's all intact. Well, you can take a patient like this, and one of those patients was described very well by a neuroscientist at NYU named Elizabeth Phelps. Phelps was able to show that a patient with an amygdala lesion had a hard time kind of seeing the world in terms of social meaning. And so as an example of this, she showed her patient with this amygdala damage the famous film by Hyder and Simmel. Now, the Hyder and Simmel film was recorded back in 1944, and the idea behind the film was you could take some inanimate drawings and make them move around in terms of animation. And there's nothing human about them, but as you watch the film, you still see a story happening. You still see some basic geometric shapes moving around on this film in such a way that it looks social. So I'll go ahead and show an example of this. So this is the Hyder and Simmel film that'll be playing as the video, but I'm also going to be playing some audio, and the audio is of 2 women who are just watching the film and describing out loud what they see. And notice as you listen to them talk, they basically are describing social behaviors. They see social meaning here in this little film.
Well, it doesn't. You'll know when it starts. There's a big black spot. Okay, there's a triangle in a box, and it's moving, and there's another triangle falling, and in a circle, and the rectangle box opened up and a big triangle came out, and the little triangle, and the big triangle's moving. It just hit the little triangle. The ball's getting in the box. It keeps pushing the little triangle. They're fighting. The triangles are fighting, and the ball's about to fall out of the box. The big triangle's trying to jam the little triangle into the box. Ball just got shot in the box. And the big triangle is chasing the little triangle, and it opened the box, and it's trying to get into the box and eat the ball. I don't know what's happening. The box shut. The little triangle is climbing the box. The ball is moving. The ball is moving. The little triangle's going to get in the box. It's opening the box. It's gonna get in. I don't know why, because the big triangle's gonna eat it. The little box, the ball's trying to get away. They shut the triangle in the box. They won. They're kissing. Now they're being chased by the big triangle. They didn't think he could get out of the box, but he can. A picture of a blackboard flashed in the back. They ran away, and the big triangle's all alone, and it wants to shut the box, but the box won't shut. It broke the box! What's that about?
Okay, so again, this film, by the way, Hyder was a social psychologist who was interested in how people ascribed meaning to stimuli and to try to kind of see social behaviors out of sort of random stuff that happens. So here, if you can again imagine that Phelps's patient who had an amygdala lesion, when that patient watched this film, they described it in much more neutral terms. They would have said things like, I see a triangle inside of a square. Now the triangle is moving up to the left and the square is opening up. Then the triangle moves to the bottom and there's another triangle, and the other triangle also moves up and then it moves back down. And then there's a circle, and then the two triangles are moving at the same time around the box. And you can notice in that kind of description, it doesn't sound like anything social going on. It doesn't sound like the triangles have a personality or they have goals or intentions, that they're trying to hurt each other. One's chasing the other or might try to eat the other one. All of that kind of stuff is what most of us who don't have damage to our amygdala would see when we watched a film like these two women that you just heard in the film. Okay, so that just shows us then how important the amygdala might be just in understanding and helping us understand social behavior. All right, the next part of the limbic system to talk about is the hippocampus. The hippocampus, you can see here, is involved in learning and memory. The hippocampus also measures or keeps track of glucocorticoids in the bloodstream. Glucocorticoids are a class of hormones that are released by the adrenal gland, mainly, to prepare the body for stressful situations. So it's basically a messenger being sent out through the bloodstream across the body that we're now in a time of stress. And so different parts of the body, different organs need to do different things as a result of this. One of those glucocorticoids that we'll talk about in greater detail in a future lecture is cortisol. And so what I'm saying here is the hippocampus actually has receptor cells in the hippocampus that pick up or bind with glucocorticoids that are in the bloodstream and then can feed back to the hippocampus basically a readout of how much that stress hormone has been released into the body. So it actually keeps track of these things that are in the bloodstream, what the levels of them are, and it particularly keeps track of them in terms of stress and what can be done to effectively reduce that stress. So what it's kind of seeing is, or it's keeping track of in its memory, is when there are certain kind of events in the body that actually cause the body to have a stressful response, and then it seems to take in information from the rest of the cortex about what was done in order to reduce that stress. So it helps us to learn how to deal with stress, if you want to put it that way. The septum is a midline structure, so it's right there in the middle, and it's much like the septum in the middle of our nose. That's why it's called our septum. It's the one that people get pierced. But here in the brain, we have a septum, this midline structure, and it primarily seems to inhibit aggression. And it has cross-inhibitory projections with the amygdala. So when it releases its neurotransmitters to the amygdala, it's trying to basically inhibit the firing of neurons there that might cause the amygdala to go ahead and project to creating aggressive behaviors. So the septum seems to be involved very much in this inhibition of aggression. The hypothalamus is really quite important. You can see that it's primarily involved in homeostasis via keeping track of what's going on in the peripheral nervous system and sending signals to change things that are going on in the body. Some of the key subparts are shown in the picture below, and you can see here we have this ventral medial nucleus. This is involved in sexual behavior, and we'll talk more about that in lecture 10. There's the suprachiasmatic nucleus, or the SCN. The SCN is involved in circadian rhythms. The paraventricular nucleus, or the PVN, makes cortisol happen. So it's going to cause a hormone to be released that will then eventually cause you to have your cortisol levels go up. And it turns out that this PVN is innervated by many different structures. So many other structures in the brain have messages sent to the PVN about whether or not basically cortisol needs to be released at this particular time. And then we have the lateral hypothalamic area. It's involved very much with hunger. It kind of pays attention or measures glucose and insulin levels. And you also maybe find it when people are hungry for information. So it's not just the literal hunger of food, but it also seems that the lateral hypothalamus might be involved when we're seeking information, that we need information. So those are all key subparts of the hypothalamus. Then we have the ventral tegmentum area, or the VTA, and the nucleus accumbens. What's important about these particular areas is that their neurons release dopamine. And by releasing dopamine, that means that they're involved in pleasure and reward. And you get a lot of appetitive behaviors because of what goes on with the VTA and the nucleus accumbens, things where you want to approach, to consume, to play, whatever it is. And very much involved with this is addiction. So addiction is very much involved in the areas of the VTA and the nucleus accumbens. These areas are also active when you're anticipating reward. So for instance, if you have a conditioned stimulus, like a light goes off, and it means then that the animal, if it presses a lever, it's going to get fed, just seeing the light come on will actually cause dopamine levels to go up because of the VTA and the nucleus accumbens. So the animal knows, like, there's that light that I've learned that it gives me food if I press the lever. So seeing that light causes a rise in dopamine. If you go ahead and give a drug, for instance, that blocks dopamine production in those areas and the light comes on, even though the animal has already learned from before that the light means press the bar, you get food, it won't press the bar. So it can't have that anticipation of reward because it's not getting the dopamine levels up. All right, now let's move on to layer 3, which is the neocortex, which we're going to spend the rest of the lecture basically on. Now, keep in mind, when we look at the rest of our brain, the neocortex, we do have these two hemispheres. They're the same structures on both sides, so we have like identical copies of things on either side. But there are some differences in function. Some of those differences are talked a little bit about in the book, but we're not going to focus too much on lateralized differences of function here. Keep in mind that in the connection between the two hemispheres is the corpus callosum, which has lots of tracks that are going back and forth. And you can see here we have lots of long tracks that go across the brain, where basically those are axons or bundles of axons that are moving from one part to a very far part to another part of the brain. We also have these long projection fibers that go from the middle of the brain up to the outer surface of the brain. And then finally, you can also have short tracks up in the upper parts of the cortex, where they don't actually come back down into the midbrain or into the corpus callosum. They're just little tracks connecting neurons in different regional areas. So those would be short tracks in the brain. The whole idea behind the brain is, one of the things you need to remember is that we have these sort of like squishy parts here throughout the brain. And the part that's kind of squishes up there in these like little tubes looking things, those are gyri or a gyrus, okay? So you can see the bulgy bits are all of the gyruses or the gyri. And then what those deep grooves are that separate each of the bulgy bits, that would be a sulcus or a sulci. So we've got sulci and gyri throughout the entire brain. And what you can see in this particular picture is it's trying to point out that we've labeled different regions of this cortex by their area and what their function is. And you can see over there on the left, we've got the frontal lobe. In the middle, we have the temporal lobe down by the ears. We have the parietal lobe that's in the blue. And then sort of in the green there, you can see the occipital lobe. One big important sulcus is called the central sulcus, which is right down there across the middle. And on one side of it is known as the precentral gyrus, which is where we have our primary motor cortex. And on the other side, we have the postcentral gyrus, which is where we have our primary sensory cortex. So the primarily what controls our movements of different parts of our body is done by the motor cortex, and where we get our sensations from, remember, from the peripheral nervous system that gets processed by the brain, that would be done up here in the postcentral gyrus or the primary sensory cortex. So there are names for all of the different gyri and all the different sulci. And I think if you were to go on and become an anatomist or neuroanatomist, or you're going to go on into medicine, you probably would need to know what all of the different parts are here that are, they're, like I said, they all have different names. We don't need to know them for this course, just need to know that we have these sulci and these gyri. So you do want to get to know your brain, though, lobe by lobe, know where the frontal lobe is, the parietal lobe, the occipital, and the temporal lobe. So these are the four main lobes in our neocortex. Another distinction that you can see within each of those lobes is like little areas that are marked off with numbers like 4, 6, 8, 9, 44, and so on. What those are known as Brodmann areas, and they were developed by Corbinian Brodmann in 1909. These areas were labeled by Brodmann just by him looking through a microscope and looking at them visually. And he didn't quite understand yet what the function of these different neurons were. He was more interested in sort of saying a bunch of these neurons have like the similar shape when they seem to be connected to one another and they kind of clump together. And so based on their cytoarchitecture, the architecture of their neurons, he was able to say that a group of neurons that went together became one area, Brodmann's area. And so you can see the numbers here kind of show you where roughly each of those Brodmann areas are. And you can see that areas 1, 2, and 3, for example, right there along the central sulcus, this is where we have our primary somatosensory cortex, where we've got, like I told you, where motor behavior is being processed, where sensory information is being processed. So even though he didn't quite understand that those areas, 1, 2, and 3, were involved in the primary somatosensory complex, they looked cytoarchitecturally similar, and that's why they became 3 distinct areas right there. Some other important areas would be Area 9, which is the dorsal lateral prefrontal cortex. We also have Area 10, the anterior prefrontal cortex. And way back there in the back of the brain at Area 17, where we have the primary visual cortex. So sometimes when you're reading articles that involve brain behavior, especially fMRI scans and so on, you'll see that in those papers, they often will talk about Brodmann's areas in terms of activation of particular areas that might be involved in a study. So they give us like very precise locations where in the cortex something is happening. Now, what we're going to do next is we're going to move on and talk about more of the cortical areas in terms of those that are most relevant to what I call the social brain. Okay, so these are areas that have something to do with the social behavior, and they have other functions as well. But in terms of us trying to understand social behavior, these are some important neocortical areas for that. So let's take the frontal lobe first and look at 3 divisions of the frontal lobes. The first one I'm going to look at here is the dorsal lateral prefrontal cortex, or the DLPFC, that you see kind of painted there in purple. And remember that there's going to be a left and a right one because each hemisphere has a dorsal lateral prefrontal cortex. The main thing that the DLPFC does, it seems to provide top-down input for task-appropriate behavior. So this is where the processing of your choices that you might have of how you could behave next, what choice you should make, how should you respond to that particular insult, and then how you're going to respond to it, not what the initiation of the action is, also is going to be done by the DLPFC. This is very much important for social behaviors. In fact, patients who have damage to the DLPFC have problems with aggression. They might increase their use of profanities and just have like a loss of drive and motivation to deal with other people. And so this really affects their social behavior quite a bit. Then we have the VPFC, or the VMPFC, which is the medial and ventral medial prefrontal cortex. This area, again in purple there, is strongly interconnected with limbic structures. So this has a lot to do with emotions that are happening from the limbic system. And we control our behavioral and emotional output, and how we interact with others is all being processed or mediated by the areas of the medial prefrontal cortex. So it becomes a very important area for social behavior. And then we have the orbital frontal cortex, so the OFC, which is indicated here in red, way down in the front, just above your eyeballs. This is important for emotion and decision making. We're going to see in a little bit later in this lecture, we're going to talk about Damasio's somatic marker hypothesis and see that the OFC is very much important for that. The patients who have damage in the OFC have a hard time processing emotions. It's involved in addictions and basically overall emotionality. Now, moving a little bit away from the prefrontal cortex to what's called the anterior cingulate cortex. So this is like deeper inside the cortex. You can see here that we have this big cingulate gyrus, okay, a big glob there near the center of the brain. And the part that's near the front, the anterior cingulate cortex, is involved in motor control, cognitive processing, and arousal. and it seems to be essential for any sort of willed control of action. So when you have a voluntary choice, that you're thinking deliberately, I want to make this choice, I want to act in this particular way, or I'm going to be on my best behavior at this funeral, whatever it is, the anterior cingulate cortex is going to be very much involved. It also seems to be involved in detection of conflicts. When you have maybe two choices here that maybe, for example, might be morally in conflict with one another, you're going to get more activity in the anterior cingulate cortex. And then we have the anterior insula, or insular. This is, again, part of the cortex, and it's just a section there you can see that's marked off in purple. It's sort of tucked down below. The insula is shown in purple. It's Latin for the word island, and it's tucked between the frontal and the temporal lobes. The insula is where the brain records bodily sensations and discomfort. If you smell a disgusting odor, you feel pain, you're being choked, you feel sad, or even if you're being excluded from a social event, you're going to see that that pain is being processed by your insula and will be active there. Then we have the inferior frontal gyrus, or the IFG. This is a posterior region of the IFG that's involved in emotional judgment, and it might have a role in top-down aspects of like emotion recognition, such as deciding what action to take on someone else's emotion, or predicting what someone else might do based on how they're behaving. So this is going to be an important part of the cortex. And then we have the temporal parietal junction, the TPJ, and the posterior superior temporal sulcus, the PSTS. This is involved in mentalizing. For example, when you are hearing someone tell you a story, or you're reading cartoons or something, and you're trying to understand the story and the motivation of the characters. Even when you're listening to someone else talk about their life and you're trying to mentalize what they might be going through, you're going to find that these areas are going to be active. So it's possibly involved then when predicting what movement a conspecific, another member of your species, is about to make. Other researchers just believe that this area is involved when we're trying to understand other people's states, when we're trying to understand what emotion they might be on. So this might be involved very much in empathy. All right, so we've just gone through these parts of the cortex that are involved in what I call the social brain. Now let's go ahead and talk about how they kind of work together. And you might remember that I said that the original problem with the triune brain is it made it seem like, the layer 3 feeds down to layer 2, and layer 2 feeds down to layer 1. But it really happens that all of them are interacting all the time, and emotion, thought, and behavior can't really be separable in terms of how these processes are affecting one another. So let's go ahead and look at just a bit how emotion might originate when we talk about social behaviors. So the better way of thinking about this is that the neocortex and the limbic system influence one another. The limbic system influences the reptilian brain and vice versa. And of course, the neocortex can affect the reptilian brain and vice versa. So they're all three interacting with one another. Now, in order to understand emotion a little bit, let's go back to one of our earliest psychologists, William James, an American psychologist who wrote one of the first big textbooks in psychology, and one of the first people in psychology to really talk about emotion in great detail. In 1885, he wrote an article called "What is an Emotion?" And he posited in this article that emotion was bodily changes that follow directly the perception of the exciting fact. And our feeling of the same changes as they occur is the emotion. So what he was hypothesizing there was that what happens when we are in an emotional situation is that our body reacts to that following the perception of whatever it is. So you have to have some sort of visual stimulation, and then your body reacts to that perception. And then once the body is reacting like that, it's feeding back to the brain, and then that is what the emotion is. Your brain processing how your body is affecting you. Okay, so you're going to need to have your visual system, of course, or auditory system to get the input, but then the input's going to be sent down into the body, perhaps through the reptilian brain, and then your body's now having a response to it, and then it gets sent back up, and it's processed now into the neocortex, and then you can actually know now that you're feeling angry or sad or whatever it was. James originally talked about this in 1885. He soon found out that there was a Danish physiologist named Carl Lange who had a very similar idea about this, and then he put them together, and it's now known as the James-Lange theory of emotion. And so that became sort of a dominant approach for a while because William James had such influence with his big textbook in psychology. And so a lot of psychologists, this is what they adopted as their theory of emotion. But then we have Walter Cannon come along, and Walter Cannon starts to critique this very dominant viewpoint of the James Lange theory of emotion. And he specifically focuses on the part of the theory that had to do with the autonomic nervous system. He thought for us to perceive something and then have our body change, that the autonomic nervous system was just too slow to have a perception happen, and then your autonomic nervous system would happen really quickly and change and then feed back up to your brain. He just said it's just too slow. Like, for instance, I told you how skin conductance activity, which is part of the autonomic nervous system, you're sweating in your skin, it just happens in seconds. So it takes a while for your body to process and change, and he just thought that's just too slow. He also thought that the problem was that the autonomic nervous system didn't have any specific patterns for different kinds of emotions. So how would you know you're angry or happy or sad? Because each of these different emotions just causes all sorts of changes in your autonomic nervous system at the same time, and therefore you wouldn't be able to know specifically, your brain wouldn't be able to specifically read from your body which emotion you have. And he also said access to our own physiological responses is poor. So he says, can you really report to me what your heart rate is like right now? Can you really tell me if your skin is sweating? So if you really were depending on your neocortex to make sense of these changes in your body, you would expect that people would do a pretty good job of reporting exactly what's going on, and they don't. Okay. So along comes Stanley Schachter, which was a theory that he proposed in the late 50s, early 1960s. And his theory had origins in this original criticism of Cannon. So Schachter was fully aware of what Cannon had said about James Lange theory. But he also was aware of a study that had been done in 1924 in which Marinon, this Spanish physiologist, had given a group of patients against their will, without them knowing, he had given them shots of epinephrine that caused their autonomic nervous system to become quite active. So they had like this sort of sympathetic discharge that got them all activated. And he noticed that he noticed that his patients, when they were given this injection, started having all sorts of feelings of different kinds of emotions. And so what Marinon and what Schachter thought was, hey, look, despite what Cannon said, if you do get a change in your body, like you have some sort of sympathetic discharge, it causes emotions to happen, just like James and Lange would have said. But what Schachter thought was missing in the original James Lange theory was that in order to have this work, you need to have your neocortex provide some context to try to understand what's going on. So he says in any emotion, there's this diffuse sympathetic charge, and that this becomes named and identified through the situation in which it occurs and the individual's perception of the situation. So he's saying that, yes, you may have like a perception of something and it causes your body to become activated. You have this diffuse sympathetic nervous system change, but you're going to then need your neocortex to kind of look at the situation and say, why am I feeling this right now? Why do I feel activated? Oh, it's because that person just insulted me. Oh, it's just because I got really bad news and now I know I need to be sad. So the emotion comes from a combination of having this feed up from your body as well as having your neocortex make sense of the situation. So he said arousal comes first, and then this is guided by cognition. So this sort of two-stage theory of emotion became the sort of dominant approach in the 60s and 70s. And then we can move along to a more recent sort of instantiation of all of this, and that would be Joseph Ledoux's work. Joe Ledoux is a neurobiologist, psychologist who's interested in the understanding of the emotions in the brain, and he's mainly worked with rats to do this kind of research. But what he has argued, and you can see this in his book called The Emotional Brain in 1996, is that his research very carefully showed that you could actually have very little cortical processing of a stimulus and yet have an emotion. And what he's trying to show you here is that there are very few neurons connecting between your eyes to the visual thalamus. And so some optic information can be sent off to your thalamus, to the visual part of where visual processing happens in your thalamus, that then projects down to the amygdala and maybe would then tell, because of a particular sort of stimulus characterization there by the visual thalamus, that this is dangerous. And so your amygdala immediately says, boom, we are in a dangerous situation, and your body becomes fearful. Okay, so it wouldn't require that you went all the way up to the visual cortex or to the rest of the parts of the prefrontal cortex to make sense of the situation, you could just have this very quick pathway that's all subcortical, that doesn't involve the cortex. And so there could be some kinds of stimuli out there that maybe through prior learning, maybe through evolutionary hardwiring, that when we see that stimulus or hear that stimulus, we immediately have a reaction that affects our, for instance, our amygdala and gives us this emotion. And it doesn't require a lot of cortical processing. So what's kind of happened as a result of Ledoux's work and other people's work in emotion is we have really sort of like this dual process approach to emotion. So when you see something out there that's going to cause you to feel emotional, you could have a pathway that goes to the cortex. And then you would make sense of this thing and you say, look at that. Oh, look at that. She just said something really insulting to me. That makes me angry. And you have an emotional experience. There could be other kinds of things, though, other kinds of stimuli or events out there in the world in which you just, because of prior learning, you're instantly able to process it. It just goes through your thalamus into your amygdala through the subcortical areas here, and it gives rise to an emotional experience as well. So we don't need to have the neocortex commenting and processing everything in order to have an emotion. It by itself, though, could be very much involved. It might even be that you're kind of sitting around and you have a memory of something that caused you to remember something that was really horrifying in your life before. That wouldn't necessarily involve the subcortical brain structures. The cortex by itself is pulling out that memory, and then you have an emotional experience from it. But there's other types of stimuli out there that we could just process very quickly without much cortical processing at all. So that leads us now to our final sort of approach or theoretical model here of this lecture in terms of how emotion might work. And you'll see this discussed a bit in Sapolsky's book. It's the somatic marker hypothesis that was proposed by Damasio in 1994 in his book Descartes' Error. And what Damasio, from working with his patients like Elliot that I talked about at the first lecture, he argued that imagined consequences are associated with positive or negative gut feelings or somatic markers. So he's saying is, as you move around in the world and you kind of think about things you could do next, as you kind of think about, should I go that way? Should I go this way? Should I go hang out with this friend? Should I go hang out with that friend? Should I respond to this text message or should I not respond to it? As you kind of think about what you might do next in terms of like an emotional, social situation, as you kind of go through that in your head, you're going to have little positive or negative gut feelings about whether or not you should do that. So you might think about this alternative and think, some really positive feeling, or you might think of another alternative and think, oh, that's negative. I shouldn't do that because you got a negative gut feeling about it. Those kind of gut feelings are called somatic markers. So where do they come from? Well, they come through learning. So in our vast experiences we have in different social situations, making different decisions. For example, even if you took the simplest thing that's not a social situation, but let's say you're at a restaurant and the last 3 times you picked this one item on the menu and it always made you feel sort of bad when you had it. Now, when it comes to the 4th time that you're there, maybe this wouldn't even require 3 or 4 times to go to the restaurant, but maybe this next time you go to the restaurant and your eye goes down and you see that item on the menu, you get that negative gut feeling, don't want to have that again. So you don't pick that, you choose something else that might sound better. So you would learn from past experiences, kind of like learning common situations that you might be in socially, or even what certain people have done to you in the past or how they've helped you in the past. You're going to have somatic markers associated with these people and these events that you use to help you make decisions about what you're going to do next. And the gut feelings themselves may be conscious or they could be unconscious. They could affect you in terms of what you're going to do next without any conscious deliberation. It makes our decision making and our choices about what we're going to do socially more efficient. They kind of act as alarm signals to warn you against bad choices and bias you towards good choices. And you might remember I told you about Elliot in that first lecture, one of Damasio's patients, who had hung out with some bad people, had lost a lot of money in a bad investment. And the idea here is that his somatic marker system had been disrupted by his tumor that he had had. And so by having some brain damage, having a lesion, it might interfere with your processing of the somatic markers, or maybe the somatic marker doesn't even come up. You don't even have the gut feeling because of the brain damage. And therefore you would make bad choices because you can't rely on your gut to help you. So the way that they've looked at this is, just to give you one example, this is like something they did back in the 90s. They would take patients who had lesions in their ventral medial prefrontal cortex or their amygdala. Either one of these areas would give to you basically the same sorts of behaviors. So one of Damasio's colleagues, Antonio Bashara, I think his name was Antonio, but anyways, Bashara was his surname. They worked with patients who had this damage, right? VMPFC or amygdala damage. All of these patients had normal intelligence. So they weren't impaired in terms of their IQ or anything, but they often made poor decisions in their daily lives. The VMPF damage prevented them from acquiring somatic markers according to the somatic marker hypothesis, and therefore interfered with their decision-making in complex situations. And the amygdala damage might also interfere with the processing of somatic markers. So that's why they kind of grouped these two patient groups together. So what they did is they came up with a really simple paradigm to kind of show how patients with this sort of damage make bad choices and they don't learn from prior experiences. They came up with this thing called the Iowa gambling task. Now it's called this because at the time Damasio was at the University of Iowa with Bashara, and so at the University of Iowa they came up with this task. And what you would do is you get 4 decks of cards as a participant in the study. If you drew a card, the decks would be turned face down, and you would pull up the first card off the top of the deck. And if it was from deck A, what would happen there is you could gain like $100 or you could lose $1,000. So this is all a game, right? So they're not going to really win this kind of money. But B would have a gain possibly again of $100 or you'd lose $1,200. But then C, you can see those decks, you could gain $50 or you could lose $75. In D, you could gain $50 or the card might be that you lose $125. So the point is that the decks A and B, they have really big gains. You could get twice as much money on chance if you picked a card from one of those 2 decks, but you could also occasionally lose a lot of money. You could lose everything you had basically if you drew a card from A or B and it had a loss on it. So again, each deck has you either win money or you lose money. And A and B, you can win big, but you also lose big. In the good deck, C and D, you have more modest amounts of money that you can win when you pull up a card from one of those decks, but you also don't lose as much. So the losses are less, right? So you would think that if you're taking turns, one at a time, you get to pull a card off the deck. You don't know any of these rules, by the way. You're just being given 4 decks and you're said, go ahead and draw a card off the deck, and whatever it says, we'll go ahead and give you that kind of money for this game. So you would draw from deck A, B, C, and D, and maybe as you kind of go through that, you think, I really like C and D. I'll keep drawing from C and D because eventually you're going to maybe win some money. You're not going to lose very much. But over there on the A or the B decks, you could, sure, win a lot of money, but you're also going to be wiped out sometimes when you pick from A and B. So the point is that a normal control participant in this study would learn after many trials to just only pull their cards out of the decks C and D, not pull them out of A and B. All right, so what happens here? Well, when you actually look at these 3 different groups, and here I have the normal controls, we have amygdala patients, and we have ventral medial patients. And by the way, the normal controls were matched in intelligence and age with the patient population. Well, the normal control participants would learn after 100 trials. Well, the normal control participants would learn by the 100th trial of this game to choose primarily from the good decks. So you can see that where it says advantageous decks C and D, that's what they're going as they go from the first trial to the 100th trial. They're picking more from those decks and they're pulling less from the disadvantaged decks. The people with amygdala damage, they never seem to really learn about this. In fact, if anything, they start to pull more cards from the disadvantaged decks than they do from the advantaged decks. The same thing happens for the ventral medial patients. They don't really pay attention here, it doesn't seem. And in fact, what happens in the end is they start pulling off more cards again from the disadvantaged decks from A and B than they do from C and D. So these patients developed a lasting preference for the bad decks. Now, why is that? Well, because every once in a while they get this hit where like they get $1,000, but overall they're losing a lot of money, right? So they're not going to end up coming out very far ahead here. So if we look at it, we can see that they're probably going to be the case that the normal control patients are going to end up with the most money at the end, and the people in the other 2 groups don't end up with very much money because they never learn which ones are the good decks. because there's some interference here with their somatic marker processing. Now, another thing that Bashar was able to do in 1999 was measure their skin conductance activity. So the measure of this autonomic arousal, right? And so what you could see is just before they're about to pick a card from the deck, you could see if they showed a skin conductance response. So it's like trial 4, make a pick. And so as they look across the 4 decks of cards and they're about to pick a card, you can see that normal participants, these normal control participants, that if they picked from deck A and B, just before that, as they anticipated thinking about a deck A and B, they would actually show a skin conductance response. But if they picked from one of the advantageous decks, they showed a smaller skin conductance response. They didn't sweat as much. So Bashar used this as evidence that that skin conductance activity, that sweating on your fingers, is part of that somatic marker. And as you sort of process this, maybe you're not even consciously aware yet of what the rules are. But anytime you kind of go over to A or B, you get this little bit more of a sweaty response, and it maybe tells you, oh, I better go over to C or D. Okay? But you can see that the amygdala and the ventral medial patients, they're not showing any difference between advantageous and disadvantaged decks. After all the trials are over, they're still likely to pick one or the other, and they just don't seem to show any kind of somatic marker whatsoever. So all the participants would generate skin conductance responses after each gain or loss. This is just talking about what would happen before, right? And so the control participants would show anticipatory skin conductance responses before picking from the bad decks, but the patients would show no anticipatory skin conductance responses. So this sort of again shows us that if you just take this simple little model of picking cards from decks, that if they're going through their everyday life and they're making social choices, they're not really learning too much about what's happened in the past because they're not able to access those gut feelings of positive and negative things like this person's good for me or this person's bad for me. They're not making that kind of learning happen because of the lack of their somatic markers. And so this kind of shows us again how important it is to actually get feedback across all 3 layers when we're trying to make emotional decisions that might involve social behavior. So just by the way, to wrap up, to talk a little bit about those somatic markers, there have been some criticisms of this research on the Iowa gambling task. So if you go back and look at Maya MacLean, for instance, in 2004, they see some sort of problems with the whole Iowa gambling task as a method. So that's something just to keep in mind. And then there's other versions of the gambling task that have been developed. And some of these involve reward decisions that are more risk advantageous. And people who don't have damage to the VMPC even do more poorly on that sort of task. So if you kind of change it around a little bit, you can find people who are not patients also making bad decisions. It just depends on how you kind of rig the game and make it more risk advantageous or disadvantaged. But overall, I would have to say that in the last 25 years, that the data are generally consistent in supporting the somatic marker hypothesis, that our mild sort of emotional responses we have as we're thinking about things in the world do play a role in our decisions and how we do things socially. All right, so that's all of the material I have for this particular lecture. We've talked about MacLean's triune brain, and again, we have lots of caveats about that and how we're going to use it as a metaphor. And then we talked about these 3 layers of the brain. We have the reptilian or brainstem that I talked about very briefly. We have layer 2, the limbic system, and there were several structures in there that are important to know about. We have the layer 3 and the neocortex, and I specifically focused on those parts of the neocortex that would be involved in the quote-unquote social brain. And finally, we ended up talking about how, as an example of how these 3 layers could communicate with one another, looking at how emotion and social behavior happen. All right, and that's all I have for this lecture. See you next time.